be here in Portugal. I must say I really love this meeting. It's got a combination of great science, exciting people, and a wonderful city. So with that, I'd like to tell you about our latest library. So what we're trying to do, our goal here is to get drug-like antibodies directly from selections. And so some of you may have heard Andre speak earlier. I'm going to use three terms. Generation 1, this is a library I made 20 years ago with Daniele Sblatero, who's here in the audience. And this was made from full VH and VL genes, amplified from 40 donors. We pooled the oligos. We didn't do any next-gen sequencing because, of course, it didn't exist 20 years ago. And the antibodies we pulled out from that were quite good. Generation 2 was essentially conceptually identical, except that rather than using pooled primers, we separated primers, and we did over 500 PCRs, assembled them together, and there was quality control using next-gen sequencing all the way through the process. And we found that by doing this, the affinities of the antibodies that came out were better than the affinities of the antibodies that came out here. One of the surprises here, as I'll show later, was that the diversity of the antibody library was far less than we thought it was when you actually measure it. And then finally, what I'm going to talk about today is generation 3. And this is a new synthetic concept where what we've done is we've taken natural CDR sequences, we've embedded them within therapeutic scaffolds, and then eliminated all the sequence-based liabilities in order to essentially improve the quality of the antibodies that come out. NGS was also used throughout for quality control and also for diversity measurement. And so we all know what a bad therapeutic antibody looks like. Bad pharmacodynamics, bad pharmacokinetics, and difficulty in manufacturing. And the converse of this is a good antibody, which has good pharmacodynamics, has good pharmacokinetics, and is easy to manufacture. So having identified these as the goal, then one thing we want is a good selection platform, which will give us these antibodies with these properties. And in addition, will allow for straightforward screening so that we can screen as many different antibodies as possible. You want to avoid screening the same antibody over and over again, which is what happens if you just pick clones and screen them. You want to have a broad paratopic and sequence diversity. It's clear the greater the diversity of the antibodies that you'll be able to screen, the more likely you will be to hit an epitope of interest. And then finally, we argue that you want an exclusive library. And so why exclusive? Well, let me just give you a short story. Some of you will have heard this already, so I apologize to those of you. There's a tale of two antibody patents. And so these two patents, Genentech against antibodies against the APO2 receptor from Genentech, that was filed in May of '97. And then antibody against the trail receptor, TR7, was filed in 2003 by Human Genome Sciences. If you align those two targets that were used, then as you can see, these two targets are in fact the same molecule. They differ by a single amino acid down here, which is a conserved change. If you align antibodies that were in these two patents, you will see that these two antibodies also have identical sequences. So how did this happen? Well, this happened because they were fishing in the same pond. They were using the same library to do their selections. And so they ended up with trying to patent exactly the same antibody. So what we believe is that you should be fishing in your own pond, and therefore you will not be sharing sequences with other potential clients or partners. So going back to the concept for the antibody, you can take antibody domains and put them on a spectrum from good to bad. And this can apply to folding, it can apply to developability, aggregation, any of these other different properties. And the historic paradigm here has been to find an antibody with a property of interest, and then to evolve that so that you increase the affinity, and if you notice that it has developability problems, you evolve those and improve those as well. I would say that what's going on currently is that people have much better libraries, they're much bigger, and so the largeness of the library is such that you can, instead of taking clones and improving them, you can just eliminate those antibodies after you've done the selection that are not developable. And so you end up with a population which is, well, is functional in terms of the binding activity, but is also developable. Now, what we are trying to do is create libraries where everything is functional inside the library, and so that everything you select at the end will be directly usable. So just looking at the general library design overview, so what we are proposing, before we go there, a couple of definitions. So you'll hear people talking about liabilities. There are essentially two sorts of liabilities, at least I'm going to talk about two here. The first is a sequence liability, and this is an amino acid sequence that is known to create potential problems. But because it is known that it will create potential problems, it doesn't mean that it will always create the potential problems. It's a theoretical liability, and it may not be a liability within a particular sequence context. For example, a deamidation site buried in the core of the antibody is unlikely to be deamidated, only one on the surface is. That is in contrast to a biophysical liability, which is a real liability that you can measure. And this includes things like polyreactivity, aggregation, stability, and self-interaction, and the like. So our general library design strategy was to first choose the best scaffolds, and we reasoned that the best scaffolds are those that are in the clinic, the best therapeutic scaffolds. There are many different therapeutic antibodies out there, and so what we wanted to pick the therapeutic scaffolds are the best. We then isolate natural CDRs, either by next-gen sequencing or alternatively by PCR in the case of the heavy chain CDR3. We now eliminate from those CDRs those which have sequence-based liabilities. And now we embed those CDRs within our good scaffolds, and producing essentially a library which has the same scaffold, but with multiple different CDRs that have been pre-screened to not contain liabilities. So, how did we choose these scaffolds? Well, Jane et al. From Adimab did a great service to the community by examining 130-odd different therapeutic antibodies at various stages of development. These are a subset of those antibodies. We just looked at the human and the humanized. And then the columns represent biophysical assays, for example, TM, polyspecificity, and various other things. So if you color code those so that you've got bad ones in red, and when I say bad, what I mean is these are biophysical liabilities defined as the 10% worst among approved antibodies, then the blue ones are the good ones. It's obvious that there is a spectrum here, but from the purposes of choosing a scaffold, we went with blue and red here. These are ranked now in terms of the number of red flags. The worst antibodies down here have 8 red flags, and the best, as you can see, approximately about a third of them, third to a half, have 0 red flags. So they're good antibodies. And what we chose was these 5 at the top that have no liabilities, no red flags. And then we also chose this one down here. I'll talk about this later. This is sirukumab. We wanted to, when we assessed developability, we needed a control to assess what a bad antibody looks like. So these are the 5 antibodies. And we chose them on the basis not just of the liability-free nature of their scaffolds, but also on the basis of their genetic diversity. So you see we have VH1, VH2, VH3, VH4, and then VH1 again. And these are coupled with kappa1, kappa2, kappa3, kappa4, and lambda. These are very low immunogenicity rates, and these are all determined in patients, and this one hasn't been done. So now, what about the diversity? So I've mentioned the concept. So why should we be using in vivo generated CDRs? Why is this better than using synthetic? Well, first is that CDRs in natural antibodies have undergone quality control. If an antibody is not expressed on the surface of the B cell, that B cell dies. And that poor display on the surface is due to a poor expression rate in the antibody and a poor display on the surface. So these are excellent sources of functional, well-folded binding loops. They also avoid covariance violations. What do I mean by that? Well, sometimes you may be able to have an acidic residue here and a basic residue here, but you have to have one or the other. And if either of them is both basic or both acidic, for example, then you have what's called a covariance violation. So here is a list of collection of CDRs. Now, comparing the two sorts of diversity, the CDR1 and 2 diversity, this is germline, and this is true for both light chain and heavy chain. And in these, germline sequences dominate. They dominate enormously over mutated sequences. And even within the somatically hypermutated sequences, you see a power distribution, which means that there are a few sequences that are found very frequently and a very large number of sequences that are only found once or twice. The diversity of CDR1 and 2 is compatible with oligonucleotide synthesis in arrays. What about CDR3? Well, CDR3 is different in the sense of its abundance range in that it's much flatter. So the generation 1 library that we made 20 years ago, if you sequence that nowadays doing next-gen sequencing, what you find is that basically 95% of the library has less than 5 copies of any single CDR3, heavy chain CDR3. So you've got a much flatter abundance range, but the diversity of the heavy chain CDR3 is enormous. It's much too large to be able to synthesize on oligoarrays. LCDR3, however, is compatible with oligonucleotide synthesis. We've looked at this. The most we can see is about 2 million different LCDR3s, and we've been accumulating a lot of data. And so this is compatible with oligonucleotide synthesis. So basically, the strategy was to use only CDR sequences from real antibodies, to use what we call replicated natural diversity for all of the CDRs except heavy chain CDR3. And these have been extracted from our data sets. We eliminate sequence-based liabilities, and then we synthesize the DNA in an array. For HCDR3, these are amplified directly from 1 billion purified B cells from at least 10 donors. Now, those will contain some liabilities, and I'll address those in a minute. So this just shows you sort of conceptually, PCR-wise, how it works. So having done a large amount of sequencing represented by these 4 genes here, we identify different CDRs in heavy chain CDR1 and heavy chain CDR2. We then eliminate those CDRs with either sequence problems, errors, or sequence-based liabilities. And then we synthesize oligos corresponding to these replicated natural CDRs, which are embedded in our chosen scaffolds. For the heavy chain CDR3, we do PCR using primers that are homologous to the scaffold of choice, and this is amplified directly from large amounts of RNA coming from 10 donors. And in fact, the number of B cells we use is the equivalent of the full circulating population of a single person. And then these are embedded within the chosen scaffold, and now we assemble all these CDRs together within the therapeutic scaffold that we chose. Note that in CDR1 and CDR2, there are no liabilities, sequence-based liabilities. In CDR3, there is the possibility of sequence-based liabilities. The same is essentially true with the light chain, except that we use natural replicated diversity also for the light chain CDR3. Eliminate the CDRs with liabilities again, synthesize the oligos, and assemble them into the scaffold. So what sort of liabilities are we eliminating? So these are just a few examples. We actually have a large list. We scour the literature to look for different liabilities. So this is deamidation, isomerization, glycosylation, and unpaired cysteines. Now, one question that often comes up is, well, if you eliminate these liabilities, then aren't you destroying your diversity? And the point is that the diversity that we have is so vast, you could eliminate 99% of all the diversity in each of the CDRs, and you would still have a diversity that is far too big to make. So this shows now the natural replicated diversity. So here what we have is the natural Cdr abundance range, which goes from 300,000 to 1, from the most abundant to the least abundant. So if you naturally amplify CDRs from population and you use that to make a library, this is the abundance range from the most to the least abundant that you will see. This, on the other hand, is the abundance range for natural replicated CDRs, which are synthesized in oligos. Unfortunately, they're not quite as good as claimed, so very often we see 1 or 2 oligos that seem to be about 10-fold more abundant than the rest. But nevertheless, the diversity or the abundance range of these is about 1,000 to 10,000 times better than if you were to go to the natural source. So this is for one CDR, HCDR1 from one library. All the other libraries are basically the same. And as I said, the distribution is 1,000 to 10,000 times better than the natural source. So what about creating the library? So what we do is we take these amplified fragments for VL and VH. We assemble into VL and VH. We then assemble these into a single chain. And then these are either used for display in yeast to look at the expression levels, or alternatively into the phage display vector for selection. This is merely for testing, and this is actually for selection. The theoretical diversity of this library is 10 to the 26, and it has 10 to the 10 transformants. So this shows the yeast display. So what you have in blue is the therapeutic starting scaffold, and you'll see that the library, which is in pink, essentially overlaps in all cases with the therapeutic scaffold. So what we have is a library of antibody fragments displayed on yeast that performs essentially identically to the therapeutic scaffold from which it was derived. Looking at the diversity of these libraries, this was our Gen 1 library. This is our Gen 2 library. This is from a single donor. This is from 15 donors. And this is our Gen 3 library. And what we have here is about 200 million measured CDRs. So this is not an estimate of diversity. This is a measure of diversity. How much clonal dominance do we have? Well, very little. You can see that the most abundant VH in the library is 0.005%. So we're not seeing large numbers of clones that are dominating this library. What happens when we do selections? So the selection platform we use is we start with our phage display library. We do 2 rounds of phage. We then transfer over to yeast. This allows us to sort for the highest affinity and also best expressing clones. And then we do NGS on the output and do machine learning to identify different clusters. The idea is to pull out antibodies with different paratopes likely to bind different epitopes in a target. This just gives you one example of a selection. So this is against GM-CSF. You see that 70% of the yeast displaying antibodies here are displaying and binding the target at 100 nanomolar. At 1 nanomolar, this has gone down to 40%. So a large number of antibodies displayed in yeast here are still binding the target at 1 nanomolar. If you look at these clusters, so this is now analyzed using PacBio. This is a heavy chain cluster here. This is light chain clusters here. You can see the 5 libraries outlined here. And what you'll see is that there are many different clusters. So you may say, well, OK, how different are these clusters one from another? If you look just at the heavy chain CDR3, then the difference between these clusters, the average difference comparing every single antibody in these clusters to every other antibody, the average difference is about 10 to 11 amino acids. These are very different in the HCDR3. What about across the whole spectrum of the antibody? So if you take all 6 CDR3s and compare them, then most of the clusters are between 20 and 40 amino acids different one from another. What about those sequence liabilities that we eliminated? So this was our generation 1 library. And you can see that 8% of the antibodies coming out from a selection here had 5 liabilities or more. 75%, 3/4 of them, have 2 to 4 liabilities. 6% have 1 liability. and 0.1% have liabilities in HCDR3 only, and 9% have zero liabilities. Now, when you compare that to this is the same selection against the same target, then now we have 3/4 of the antibodies coming out have no liabilities, no sequence-based liabilities. 22% have one or more liabilities in HCDR3, and this is because we've taken the HCDR3s from a natural population. And then the rest can essentially be neglected. What are the affinities of the antibodies coming out of this platform like? So this is all against GM-CSF. So you see 2 nanomolar, 650 picomolar, 900 picomolar, 2.6 nanomolar. These are all measured with the Cartera LSA platform, and these are just showing a selection of different targets, and you can see that the affinities here are all rather low, and what one would be very happy to be pulling directly out of a library from the start. When we plot these on an isoaffinity plot, you can see that there's a number here between 100 picomolar and 1, a few more between 1 and 10, and then a few between 10 and 100. And we can do that. We've done this with other antigens, and you can see that the same pattern of affinities is occurring throughout the selections. And if you look at the KDs, then essentially what we have is 20% of the antibodies are subnanomolar, and 40% are between 1 and 10, meaning 60% of the antibodies coming out of this platform are less than 10 nanomolar. What about the developability? Because this was one of the things we tried to build into the library. So what we did was we took the single chain domain, we converted that into a full-length IgG. We had this expressed, and then we tested this in vitro for various developability properties. So the properties we looked at were the purity and proteolysis, the expression levels, stability, polyspecificity, self-interaction, aggregation, and then long-term stability using 2 different methods. We either kept the antibody at 37 degrees for a prolonged period, or alternatively, we froze and thawed 10 times. Now, these were carried out using these different assays, and the antibodies that we were using were sirukumab, which, as I mentioned before, is the bad antibody. It's an antibody that's out there against IL-6 that has seven different liabilities in it. We used the parental scaffold antibodies for each of these to provide us with a baseline, and then 4 selected clones from each library. So this, just to remind you, sirukumab down here, bad antibody. So if you now look at these antibodies, and this is now looking for freeze-thaw cycles, so we have 10 freeze-thaw cycles here. The parental is in blue, and the light blue at 0, and dark blue after 10 cycles. And you can see that essentially the parental is overlapped. There's no difference. If you look at sirukumab, it has a very aberrant degradation peak. It also has a broad distribution, probably indicating the presence of aggregates. And finally, you've also got aggregates here. Now, if you look at the clone, this particular clone here, which is in yellow and orange, essentially it overlaps with the parental antibody. And the same is true for other clones coming from this library. And we see similar patterns for the other four libraries with clones tested against them. So just to summarize this library, so this is sirukumab, where we have a light blue, this is a mean property, whatever that property is, which is consistent with the therapeutic range. If it's blue, then it's better than the parental range, and if it's red, it's worse than the parental range. And so you can see sirukumab is pretty bad. Abrolumab, as you'd expect, is all light blue. That's our standard. And then the 4 clones coming out of this library, all of them are blue, and some of them are dark blue. So if you look at the antibodies, so look at all the antibodies we've tested in this way, then we've essentially tested 19 antibodies. 84% of these, 16 antibodies, have no liabilities in them when measured using these biophysical assays. 3 of them, 16%, have 1 biophysical liability, and none of them have more than 1 liability. And so our conclusions here is that we've produced this platform which does generate a high proportion of drug-like antibodies. 84% have no liabilities, the rest have only 1. 60% of these selected antibodies are high affinity, 10 nanomolar or less. We have very high antibody diversity against different targets, so we see, depending upon how we do the selections, between 500 and 5,000 different clonotypes. Interestingly, different targets prefer different libraries. I haven't shown the data for that, and that probably reflects something to do with the inherent properties of the antibodies. And then finally, our libraries are provided exclusively. This is the fantastic team that's been working on this, quite international, as you can see, very diverse. And so if you have any questions, please visit us at specifica.bio, email, or alternatively, Andre, sorry, I should have mentioned Andre was the one who built the library. He's done a great job with doing that, and he has a poster up at 67, and if you want to visit our website and so on. So I'll take any questions. Thanks very much.